Hi, everyone, and welcome to the Virtual Bite of Science. We're thrilled you can all join us today. The Teacher Enrichment Program, sponsored by the Center for Excellence in Education, which is a nonprofit founded in 1983 by Joanne DeGennaro and Admiral Rickover, father of the nuclear navy and civilian uses of nuclear power. Our mission is to nurture high school and university scholars to careers of excellence and leadership in STEM. And we offer 3 cost-free programs in the United States, which are the Research Science Institute, the USA Bio-Olympiad, and the Teacher Enrichment Program. RSI is a highly competitive 6-week summer science and engineering program open to rising high school seniors. It combines on-campus coursework with scientific theory with off-campus work in science and technology research. This past summer, RSI was hosted virtually very successfully, and we are looking at hosting it again virtually this upcoming summer. Participants from RSI do well in competitions like the Regeneron competition and often go on to secure advanced STEM degrees. Also run the USA Bio-Olympiad, which is open to any high school student. A series of examinations testing knowledge of biology concepts. The top 20 from the various tests attend national finals for 2 weeks in July. And we'll see how things play out this year. The top 4 from the national finals go on to the International Biology Olympiad. She's supposed to be in Lisbon, Portugal this summer, but we'll see. So there are free student and teacher resources that are great if you teach AP or IB biology classes. Registration actually just closed. I did not look at my slides closely enough before this event, and the exams are beginning early next February. But certainly, again, we send out information to teachers about what those application deadlines and how to get involved. We are with the Teacher Enrichment Program, and we are looking to provide opportunities to middle and high school teachers to connect with leading experts in the industry and academia, explore cutting edge research and development, and hopefully make some meaningful professional links with STEM professionals that benefit you and your students. This year we've gone virtual with our Bite of Science. I am very excited to someday go back out and travel and see all of you in person. We actually this past summer reorganized our lab bench into subject areas, and we're very excited to be working on a new website for our organization, which the lab bench will no longer be a whole separate website, but it will actually be part of the page that you go to when you want to find out information. And again, certainly, and especially, working on public-private partnerships, trying to get you've got teachers connected with STEM professionals. And especially with the virtual, please reach out to me. I have a lot of people who are very interested in talking to you and your students. And with the virtual, it's actually all easier than ever. So this is the reorganized lab bench, as you can see. So we have life sciences, design and engineering, physical sciences, technology, mathematics, and a couple of other ones. As an example, we'll click into the physical sciences drawer. And that's what it looks like. So you have across the top are science, environmental science, chemistry, physics, and space science with the presentations and associated activities and resources all together in one piece. So hopefully it makes it easier for people to find stuff on the lab bench. And we're so thrilled today to have Dr. Andrea Beyer, Assistant Professor of Biology at Virginia State University, and Frank Hatten, Education Relations Specialist with Boeing, here to present and talk to you all today. So with that, Andrea, you can take it away.
Thank you very much. And it's great to be with you guys this afternoon. I titled it From Farm to Phage, and that's mostly because I'm a farm girl and I really never expected to be a research scientist teaching at an undergraduate university. So I wanted to share with you today a little bit about my journey, how I got here, about some of my research using bacteriophage, which are viruses that infect bacteria. So as I mentioned, I grew up on a farm in West Central Pennsylvania, and growing up, it was really encouraged for me to be a housewife. And if I had any sort of profession, it was expected that I would do something more feminine, such as go into nursing or be an accountant. And I honestly, as a kid, wanted to be a truck driver. I grew up with a lot of men in my family. They were either farmers or some of them drove coal trucks in the region. And so that's something that I just thought was really cool. But the longer I was in the farm area and the longer I was in high school and doing different things, and I also heavily participated in Girl Scouts, I started being exposed to different things that made me realize that I think I wanted to do something different. I wanted to do something more. But my farm origins really did play a part into where I am today. But just a couple of pictures here. We raise cows, pigs, chickens, and all kinds of stuff. We still have the family farm in Pennsylvania today that my dad runs. What really made me like science, though, as I mentioned, was through my education, middle school, but most heavily in high school. The teachers that I had in my life were the people that influenced me to go into science. And I saw a lot of overlap, especially when it came to infectious diseases. As you can imagine, living on the farm can be a dirty place. There's a lot of germs, and there were a lot of instances of viral outbreaks where we had to vaccinate or take care of our animals when they became ill. And so that leaned me more towards thinking about microbiology. But what did I know about it? I knew nothing. And I sure did not know anything about going to college because my parents both went to high school, but never went on to further their education. So middle school, I had two, I'll just say crazy science teachers that really were fun and made science exciting and interesting, Mr. Smith and Mr. Kosmak. And I say crazy because they did things out of the ordinary. And some of them I have on this bullet point here, but I remember that they had us do these things that we would look at them and be like, why, Dr. Kosmak, are you having me do a commercial about this planet and trying to encourage people to come and live on this planet? We had to look up planetary facts and compare it to Earth. Just really fun, silly things, but it really made science exciting and interesting and allowed for us to be very creative. But really, it was in the high school that I had a biology teacher who had us out doing things that I could see easily fitting into everyday life. Probably the one that really sticks out to me was a stream study. We went out to our local hunting and fishing club and collected water samples and did all sorts of analyses. And then we were able to plug that information into Pennsylvania Conservation website, and they were collecting that to look at stream quality. And she explained about all the different parts and aspects about acid mine drainage from the area, how we salt our roads in the wintertime, how that affected the things living in the stream. And I just thought that was so cool. And some other things that she really influenced me in towards thinking about college was she told me about her time at Penn State getting a master's degree. And it really made me realize that women could do science. You know, most of the time we see men depicted in textbooks and different places for science techniques and learning new inventions and technology. And she really made me realize that women could do science too. So that's what got me started into pursuing a career path in the sciences with biology as my focus. And I had no idea what I was doing. As I said, I'm a first generation off the farm girl, so nobody in my family had gone to college before. Really didn't know how to apply financial aid because we did not have a lot of money. We certainly didn't have money to afford college, so finding scholarships, that was a big thing that it was scary. And like I said, I didn't know what I was doing, and neither did my family. My mom was really instrumental in kind of going out there and pulling resources to help me along the way, as well as Mrs. Stifler, that biology teacher that I just told you about. And then some other family members who kind of told us their bits and pieces of experiences and what they knew. So I ended up enrolling at an all-women's college in Allentown, Pennsylvania called Cedar Crest College. And what really drew me there was they had a 4-year program in genetic engineering. Back in the late 1990s, early 2000s, genetic engineering was one of those buzzwords, and it just sounded very interesting and exciting. And it combined biology, which I loved in high school, as well as chemistry that I did very well in. And it was applying it to this new field of cloning. You probably remember about Dolly the sheep when she was cloned. Things like that that really got me excited. So I did 4 years at Cedar Crest, and what really helped me pursue where I am today is the fact that they had a freshman research experience there. So my very first semester at this campus I was doing research with viruses, viruses called bacteriophage that infect bacteria. And I'll be telling you guys more about those later, but this is really where my research pathway began. It was part of a class. I had to do some research. And so I got started with these bacteriophage, and I did research for 3.5 years out of my 4 at Cedar Crest, which is kind of unheard of. And I realized I liked research so much, I did 2 internships along the way. One was with the New York State Department of Health shortly after 9/11. So right down the hall from when they were doing anthrax testing. I got to be there around that time. And then also I did an internship closer to home doing breast cancer research, because at that time I was like, maybe microbes are not for me, maybe I'll do something more molecular biology and genetics. So I had 2 really great internship experiences that influenced that I really like to do research. But also along the way, I started doing some teaching to fellow undergraduate students. So I started thinking about teaching, and that really became more evident when I graduated from undergrad and decided I wanted to become a doctor. I wanted to be the first PhD in my family and become Dr. Beyer. So I went to graduate school at Penn State College of Medicine in Hershey, Pennsylvania. If you can't tell, I'm a deep-rooted Pennsylvania girl. And in graduate school, I got my PhD in microbiology. And a big part of my training there was I got a lot of experience teaching medical students, other graduate students, and high school students, and realizing how much teaching was an important part of my life and something that I really wanted to embed in my career. So while at Penn State, I did my research on viruses related to HIV. These are called retroviruses, doing a lot of genetic studies with those. So after I got my PhD, the big question is usually what next? And so for a lot of students such as myself, when you're not quite sure where you want to go, you continue on in a postdoctoral position. And I was really lucky to be afforded an opportunity to do what's called an IRACTA postdoctoral fellowship. IRACTA is an acronym, stands for Institutional Research and Academic Career Development Award. And what that mouthful essentially means is that it's a 3-year postdoc opportunity where you're doing research, which is what you typically do in a PhD postdoc in biology or biological sciences. But I also had formal teacher education, meaning I was trained to be an undergraduate faculty member. So you can kind of see my brief timeline here. In year 1, I actually took graduate level classes to become a professor. Year 2, I focused more on research, but I started doing teaching. I had a teaching internship with our partner school. And in that time, I was at Virginia Commonwealth University for this IRACTA fellowship, so in Richmond, Virginia. And our partner school, fortunately for me, was also in Richmond, so down the road at Virginia Union University, or VUU. And then my 3rd year, I finished up doing, focusing mostly on research and mentoring. I liked my research so much at VCU. I was working in a intracellular bacterial laboratory with Jason Carline. Even after my IRACTA time in the teacher training officially finalized or finished up, I continued in the lab for another 2 years, just doing research and publishing some papers. But during that time in those 2 years, I wasn't doing as much teaching. I realized teaching is just a big part of my life. So despite having done 15 years of being primarily a research scientist, it was time for me to step into a teaching role. And that brings me to where I am now. I'm an assistant professor at Virginia State University. So this is an HBCU located just south of Richmond, Virginia. We have about 40,000 primarily undergraduate students. And you can see here a picture of our beautiful campus in Petersburg, Virginia. If you can see in the picture, there's a water tower kind of back in the distance. My building is located right in front of that. And I started there in 2016 as an instructor, and within a year I was able to apply and get accepted for the tenure track assistant professor position that I now have in biology. So hopefully by now, with that brief background, I give you an idea of how much research has really molded who I am as a person. It gave me a lot of experiences and appreciation for all the teachers along the way that have really given me opportunities to try new things and to try to see where my interests are. I think that's something so important with our students, whether it's high school or undergraduate, is to give them opportunities. And I like to say this a lot, but to try before you buy. Try it out, see if you like it. To do something, do a volunteer opportunity, do an internship. Is it something you like, or is it not? Especially as students consider going to college or further on in their education. College is not a good fit for everybody. And so sometimes it's a really great thing for students to go out and volunteer or do internship opportunities to make sure that when they commit to that education program or that experience, they're committing for the right reasons, because they're going to be more successful if their heart is in it. So speaking of where your heart is in it, my research now comes back full circle to the research I started as a freshman at Cedar Crest College, so way back 21 years ago now. And it's doing research with these things called bacteriophages. So if you're not familiar, a bacteriophage is a virus that infects bacteria. The word, or the ending of the word bacteriophage, the phage part refers to eating. And so this essentially means bacteria eating virus. And just a kind of a rough description of what they look like. When we say bacteriophage, we're usually talking about a generic virus structure that looks something like this. So under an electron microscope, you would see this kind of spaceship looking microbe. It has this head portion or capsid, and that's where its genome is located. A lot of the viruses that I work with have a DNA genome, but some can be RNA. And then they have this tail. Some of the tails can be very short, some of them can be long, and there are some bacteriophages that don't even have a tail at all. But there's this tail, and then there are these tail fibers that extend off of it. And these tail portions are what allows for the virus to specifically interact with its host. So if you think about that kind of spaceship idea, the spaceship has to find a good place to land. And so its landing spot, or where it's going to dock, are receptors on the surface of a bacterial cell. So bacterial cells have all kinds of proteins and receptor on their surface to allow for them to exchange with their environment. Well, bacteriophage, just like all viruses, they make use of that by binding to specific receptors. And once you have that interaction, it's like a lock opening up, or a lock opening up with a specific key. And so the virus can then gain entry. And viruses, regardless if it's a bacteriophage or if it's a virus like the one that's causing COVID-19, their main agenda is to get into a host cell and make tons and tons of more viruses. And that's the exact same thing for bacteriophage, except with these viruses, they are not a threat to humans. They will not infect our cells, they will not infect animal cells. These are predators of bacteria only. And here's a really cool electron micrograph picture where you can see a single bacterial cell in this like blue color. And this poor bacterial cell is being overwhelmed with many, many bacteriophage shown here in green. And so these are tailed bacteriophage. You can see their nice big green globular heads, and then you can see their tail portion, and then their tail fibers are doing the part that's interacting with the surface of the bacterial cell. And so after they have this interaction on the surface, the next step of the infection is that the virus kind of acts like a needle and syringe, and it injects this genome or this DNA through its tail and into the bacterial cell. And once that blueprint is in the bacterial cell, the bacterium is taken over and it is now turned into a virus making factory where the bacteriophage makes many, many copies of itself, and it eventually bursts out, killing the bacterial cell. So some fun phage facts, and please excuse the silliness. We always do fun stuff like this with the phage. Instead of having an F, we'll put a PH in there for phage. So fun phage facts. What most people don't know, especially if you're not familiar with bacteriophage or never heard of them until now, they are the most abundant microbe on the face of the earth. And so the estimate that is out there based on different studies and calculations is that there are 10 to the 31st power of these individual virus particles existing on the earth. And that's a ridiculous number. It is 10 nonillion, which I didn't even know that there was words to describe this beyond billion and trillion. It's 10 nonillion, or 1 with 31 zeros. That's hard to kind of fathom that there are many of those viruses out there because they're microscopic. We can't see them with the naked eye. So bear with me. If you're not a bug person, I apologize. But imagine if these viruses, instead of being microscopic on the nanometer scale, imagine now they were the size of like a June bug or some sort of beetle that you're used to seeing a lot of. So if these viruses were the size of a beetle, to give you an idea of how many of them there are on the Earth, the Earth would be covered with several miles deep of these beetles with 10 to the 31st power of them on the Earth. And while that's quite a disturbing image to fathom, It just gives you an idea now that there are so many viruses out there, and not only that, but there are many of these bacteriophage. So they're in the dirt, they're in the soil, they're in the oceans, they're in streams. They are anywhere that bacteria can be found, including in our own bodies. And interestingly enough, all of these phage that exist within marine sources, especially in ocean waters, they get the viruses are killing off 40% of all the bacteria in those water sources every day. So this is a predator-prey relationship that results in high turnover of the bacteria. So the viruses infect, kill the bacteria replicate very quickly, they make more of themselves, and it goes on and on and on. The really interesting part, though, is that there are all these bacteriophage that exist on the face of the earth, yet we really don't know a whole ton about them. There are a few phage out there that are considered the canonical, or the ones that we reference to a lot. For example, phage lambda. It's a bacteriophage that infects E. coli. That's one of the most characterized and studied bacteriophage out there. But that's one out of potentially 10 to the 31st virus particles out there, many different types and varieties. We just don't know much about them. And for the most part, we don't know what their genetic or their genes account for. We don't know what those genes encode for. Now, over time, bacteriophage have contributed to multiple discoveries in biology. I'm not going to go over these, but things that we now know as part of central dogma and part of the basics or foundations of biology, including genetic mutation, the fact that DNA is the genetic material. That was done with the Hershey-Chase experiment using bacteriophage in the 50s. And a lot of new molecular techniques, including the CRISPR-Cas system that is now being developed and used for healthcare purposes, that was developed using bacteriophage. So they've been a really useful tool over time. And funny enough, while the tool has been in the laboratory, not many groups have sat down and actually focused on the actual bacteriophage and all their capabilities in terms of genetics and their functions. So I already told you that bacteriophage don't cause human disease. I shouldn't say don't cause human disease. They don't infect human cells. They are primarily predating on bacteria. So why do we care about them as human beings, and how could they relate to healthcare? Well, they do infect bacteria that can cause human infections. And so there are a number of bacterial diseases out there. Some of these, fortunately, are not as common nowadays. But there are a number of diseases out there that you may be familiar with, that it's not the bacterium itself that's making us sick. It's because the bacterium is infected with a virus. And that virus, as part of its genome, has a toxin gene that, whenever it is expressed inside the bacterial cell that it's infecting, causes the human disease. So this includes diseases such as cholera, different types of hemorrhagic diarrhea, so some types of food poisoning associated with like salmonella, that gets onto our lettuce, for example, and they do all the recalls, botulism, and as well as diphtheria and scarlet fever. So those are all bacterial infections, but those bacteria themselves are carrying an infection inside of them that makes us even sicker. And yes, you might have realized botulism on there, or the botulinum toxin is listed on this chart, and that is carried by Clostridium botulinum bacteria. And again, it's not the bacterium that's producing the botulism toxin itself, it's the virus infecting it that is expressing that. So Botox is when they isolate that toxin from the infected bacteria and then use it for the muscle paralytic as part of our cosmetic procedures. So the bigger picture now, though, as to why study bacteriophage, and besides the fact that it's been a really great research tool over the years, is we're now finding that it's a really good tool in infecting or causing infections of bacteria that make people sick with different antibiotic-resistant bacterial infections. We call this phage therapy. And so essentially how this works is that if you had a bacterial infection, we would typically go to the doctor and get an antibiotic. Well, you're probably very familiar with the fact that some antibiotics are no longer useful or no longer effective against certain treatments or treating certain types of bacteria. And so one alternative idea is using phage therapy, where you have specific viruses that are able to infect these specific bacteria that are making you sick and be able to clear up the infection without causing the side effects that are common within antibiotics. Because antibiotics, when you take that, it's not only harming the bad bacteria causing your infection, it's also harming all the good microbiota of your body, the bacteria that live naturally on all your orifices and all your mucosal membranes, including inside of your guts. And that's why sometimes there's the nasty side effects of upset stomach and diarrhea, is that the antibiotic is killing those good bacteria. But with something like phage therapy, you'd be using specific viruses that target only the bad bacteria, and that would leave the good ones alone. So phage therapy was actually started being used right after World War II, but along came antibiotics and it kind of got pushed off to the side. But now with antibiotic resistance, there's a lot of surge and interest now in phage therapy. So why study bacteriophage? I already told you they're the most abundant microbe on the face of the earth. And what's interesting though is if you look into a lot of genetic databases in terms of sequencing for DNA, there are more sequenced genomes from eukaryotes, so plants and animals and other higher order species than there are bacteriophage, even though these viruses are incredibly abundant. And what's more, as I mentioned before, is that more than 70% of their genes have unknown function. And so there's a lot to be discovered here. What if those genes could be the next therapy for cancer treatments, or better uses for clearing up bacterial infections, or designing better products to target bacteria? As I already mentioned, CRISPR-Cas is the now system that's being used to prevent the production of certain gene products, and that was discovered using bacteriophage. So there's a big push now to learn about these bacteriophage that are so abundant and do DNA sequencing to evaluate their genes. So that comes in with my research. The program I'm associated with is a nationwide program through Howard Hughes Medical Institute, and it's part of their umbrella program called the SEA, S-E-A. It's a Science Education Alliance. And under the SEA specifically, I'm part of a group called the Phages program, appropriately working with bacteriophage, but the phages part stands for Phage Hunters Advancing Genomics and Evolutionary Science. And so this is what I now bring to my students at Virginia State. And the whole purpose of this program is to find novel viruses, characterize them in a laboratory setting using basic microbiology techniques, and then isolate and sequence their genetic material so that we can then look and compare viruses from different groups and different locations to one another so that we can learn to understand the diversity and start looking at some of these common genes within these phage groups, and hopefully start to understand what are these unknown gene functions that they carry within their genomes. This is just a diagram showing the family tree of some of the viruses that have been discovered so far with the C phages program. The colored bubbles represent family groups, just like a family tree. And so the closer together the branches are, the more related the viruses are. And hopefully just from this colorful schematic and all of the branches going everywhere, you can appreciate that there's a lot of diversity as well as a lot of similarity within these phages that we're finding. So here's some pictures of my students hard at work in the laboratory. And what I like to do is take my students at the freshman level, so straight out of high school with zero laboratory experience in many cases, and I give them basic microbiology techniques training, learning sterile technique, micropipette use, math calculations for calculating how to make different solutions. And they get to do a research project where they discover their own virus. So they have to go out and collect dirt samples from wherever, and they bring that sample back to the laboratory where we weigh it, we isolate bacteria and virus samples from it, and then we begin this characterization process and try to isolate the virus. So the names of the viruses you're gonna see here today were all named by my students. I started this program in 2018. And like I said, I work primarily with freshmen, and it's a wonderful experience because they get so excited when they realize that they are able to do real research the first year of school within the first couple weeks of their freshman semester. And it's really amazing to see how they come in thinking that they cannot do this, And then by the end of the semester, they're super confident, they're excited, and they just have a bigger outlook in terms of what they're able to do in science. Since I started the program, I've only had female students. That's not by any selection on my end, it's just the way it happened. When the students matriculate, they get to choose to sign up for the laboratory. So my first year I had 10 students, in subsequent years I have about 12. And we work pretty closely together in the laboratory when COVID is not happening, but I'll talk a little bit more about that in a minute. But you can see them all here happily at work, and they wear lab coats and they're wearing gloves. They're doing real science stuff. They have to keep lab notebooks, and I have a small research space where they get to do all this awesome work. So here are some examples of their research that I wanted to share with you. This is from my first year of the program that I had 10 students. We worked with a soil bacterium that you can find outside in your yard. It's called Mycobacterium foliorum. And the program specifically looks with a lot within this family of bacteria called the Actinobacter species. And it's largely soil and water, so very commonly found bacteria. And so the main goal was to find new viruses that infect this particular bacterium. So the students had to learn to culture the bacteria, prepare the food to make it grow and make it happy. And then they would take their dirt samples, purify away all the other stuff, and then see if they have viruses. And I don't think I have a picture here. I must have took that slide out for the sake of time. But what they're able to do is to grow their bacteria on a culture plate, so a Petri plate. And the bacteria, when it grows, it makes like a cloudy coating over the entire plate. But when there's viruses there, they see these like little spots or dead zones on their plate, and that's where the viruses have essentially exploded out and killed all the bacteria. And when those students see the plaques, they're like, oh my gosh, we discovered virus, and it's so exciting. So from the 2018-2019 cohort of students, we found, I believe, 10 viruses that year, and we were able to isolate DNA and send off 3 of them for genomic sequencing. The students also prepared electron microscopy grids. That's something I didn't even get to do until grad school, so they get to do it as freshmen. And here you can see electron micrograph pictures of their viruses that they discovered. So these little lollipop figures here, these are 3 different viruses, Owens, Teddy Boy, and Sanzafet. And then that all happens in our fall semester, and it continues if the students choose to the spring semester, where we do a bioinformatics computer-based part of the class, where they analyze their DNA sequences, and they do what's called annotation. They try to assign where genes begin and end, and they kind of put together the whole picture of the genome and how it all works together for the virus. And then the students actually publish that. So if you go on to the publicly accessible NCBI GenBank, and you were to type in Owens, Teddy Boy, or Sanzafet, you would see the genomes of these viruses now published there with the students' names listed. So this is really exciting stuff that we've been able to do with our students. Oh, here's an example of that plaque picture I was telling you about. So here's a zoom in of a bacterial plate. The white cloudiness is the bacteria growing, and where the students' viruses, you can see these white spots or these clear spots here on the white background. And those clear spots are, again, places where the virus has killed all the bacteria, and it's literally a dead zone. And then here's the electron micrograph for Teddy Boy. We have a database called phagesdb.org. Anybody is able to go in there and look at that data. I'll show you that website on my last slide. And you can see all the information about the student who discovered the virus, where they discovered it. There's GPS coordinates. So we collect all that information, and it's now publicly accessible. So many groups across the country can now use our virus as part of their studies and learning about these genomes. And so here's just an example of a genetic map that we made showing the color blocks or where the genes are located for one of our viruses. So this is Teddy Boy. This is the database that I was telling you about that anybody is able to access. So you are able to access this if you wanted to share this with your students or look up viruses. It's called PhagesDB. Or if you were to just Google CPhages database, you would probably come across it. It's phagesdb.org. And again, it has all of our viruses, not just from our school, but all the colleges across the US that are participating. This is an older picture, but it has a keeps track of how many viruses have been sequenced so far as part of this initiative. And there's tons of genes that we're discovering along the way and seeing how related they are to other viruses out there. All right, so I think I stopped there because I could easily talk about this for a long time because I really love what we're doing with the phages work. The students really like it. Unfortunately, with COVID, we're doing primarily the just the bioinformatics part with annotating genomes because that's the easiest to do online as opposed to trying to do the dirt samples. So I'm hoping for the fall we can get back to it and discover some more viruses. But I'd be happy to take any questions from any of you if you have any.
Yeah, we do have some questions. So the first one is, in terms of what we know about bacteriophage DNA, I guess how much do they have in common with eukaryotes?
Okay, so in terms of eukaryotes or viruses that infect eukaryotes, I don't know if anybody.
So like how much, like, you know, we have for like bacteriophages that have like DNA genetic material, like do we have any common genes and any of that overlapping?
For the most part, no. So viruses in general, whether it's a bacteriophage or a virus that infects a person or an animal, viruses for the most part are considered a parasite. And so when they infect organisms, they travel pretty light. So think of them as bringing the smallest suitcase possible, and they only have with them the genes necessary to make more virus particles. And so on that note, they don't have any machinery or mechanisms or metabolic properties themselves. They rely solely on the host for that. So in the case of bacteriophage, they're relying solely on the bacterial cell to provide the ribosomes and nucleotides and amino acids and everything it needs to make new viruses. So in terms of genetic content, I would say the limitation to the similarity between like us, for example, and viruses is that there's some similarities between some human viruses and bacteriophage. But in terms of eukaryotic genes, there are very few. If anything, the bacteriophage most closely resemble their hosts, which are the bacteria. And so sometimes we'll see some bacterial genes that are tucked inside the virus genomes that sometimes the viruses pick up and take with them when they leave one host and go to infect another. So very few similarities with eukaryotic organisms, but they do have some more similarities to their hosts. And then in terms of similarities to like human viruses, about the only thing that they may have in common is that capsid structure that I showed you is kind of like the head part of the virus that holds the DNA inside. There's some similarities between that protein with, for example, herpes viruses, which are also DNA viruses that infect humans. But other than that, that's probably the extent of the similarities.
Awesome. And then, so another question is, in terms of turning bacteriophage, like having phage that fight bacterial infections, how does one go about doing that, like creating those bacteriophages?
Yeah, so essentially you have to go looking for them. And that's the harder part with phage therapy. And what's being done now is that there are now institutes across the United States, I believe even the US Army is trying to build a bank of viruses that they can then put in the freezer that have been characterized by groups such as ours, that then they can have kind of on supply in case there was a patient or somebody who came down with an antibiotic resistant type of bacterial infection. So if you were to Google phage therapy, there's some really inspiring stories out there. The one that's close to home with us at C-Phages is that there was a young girl in, I believe, the UK. She had cystic fibrosis, and she had an infection with an antibiotic resistant bacteria that it was going to be lethal. She probably would have died from it. And then doctors from the UK reached out to the C-Phages program. Hey, this is the type of bacteria that causes this infection. Do you have any bacteriophage in your freezers that have been discovered that couldn't kill this infection? And they did. So they had several different viruses that were in the freezer from the C-Phages program. They sent them to the UK and made a cocktail drug, which they delivered to the young woman in her hospital intravenously. So they injected it into her, and it saved her life. And there's a couple cases of that now happening here in the United States as well. It's not FDA approved yet, but it's getting closer to being there. There's a really great book about it. It's called The Perfect Predator, and it's written by Stephanie Strasty. Her husband was infected with an antibiotic-resistant strain of bacteria while traveling, and he almost died as well, and they used phage therapy to save him. So there's a big push now to discover as many bacterial phage that we can and put them into different databases, put them into freezers in the event that if there is an infection or bacterial diseases that are caused by similar viruses that we can now, or excuse me, bacterial infections that could be, that could use similar viruses, we now have them on hand.
Continuing on that theme, is there any significance of the lysogenic phase in the bacteriophage in the treatment of multi-resistant bacterial infections?
Yeah, so for the most part, lysogenic refers to when the bacteriophage infect a bacterial cell, what we call it a temperate bacteriophage. It has an option of kind of going into like a hibernation state where it inserts its genome into the bacterial chromosome. So most viruses, if they're considered lytic, they go in, they infect, they make more virus, and then they kill the bacterial host and they release all those new virus particles into the environment. With a temperate bacteriophage, they can go into this lysogen state, and that's the state that causes those human diseases that I was telling you about, where the bacterial cell itself is infected, but it's kind of infected more silently because you don't see actual virus particles inside the bacterial cell, you just see their DNA. So in terms of phage therapy, we largely want to destroy and kill the bacteria. And so there's a greater need for finding more lytic bacteriophage that we know that we could give potentially to a human being, have them infect their bacteria and then kill them. Because we don't want to have bacteria that are loitering in the body that could still persist and cause a prolonged infection.
All right, we have a question here that asks, are there course requirements for the C-phages program?
Interest in biology. That's a lot of my students coming in, they have a variety of backgrounds in biology. Some of them have had wonderful training, but I pretty much assume that they don't know anything other than that they are concurrently taking a Principles of Biology or Introductory Biology lecture. And so I try to time it so that when we're hitting on things like central dogma, talking about DNA to RNA to protein flow, when we really get to that in C-phages, hopefully they've at least heard about it or started talking about it in class. So I try to build on what they're getting currently and not basing it on anything that they've learned before. Because a lot of students will come in and they're just like, oh, I never talked about viruses before. I've never worked in a laboratory before. They don't know what a micropipette is. So this is pretty much you can walk in just with an interest, and I will teach you everything you need to know so that you can discover a virus and hopefully get some DNA out of it.
Awesome. Okay, one last question. How many phage strains have been discovered in soil? And then the follow-up to that is kind of, are they similar to each, do you find that they're similar to each other?
That's a great question. So within the program itself now, I mentioned about going on to Phages DB. You can kind of see the snapshot of how many viruses have been discovered. And I haven't looked recently, but we're now getting into the thousands, which is super exciting because when the program started, I think there were maybe 100 or something. So we're definitely discovering more. And what's really exciting is we're finding family groups of these viruses, and they're being organized into what we call clusters. So those bubbles I showed you, that colored bubble map, our family tree, we're finding more and more branches on that bubble map. And we're finding that some of the viruses are extremely similar to each other. And then there are some that are extremely different. And if sometimes they're so different, they're not related to anything we've seen before. And we'll call them a singleton, like a loner or something brand new that we haven't discovered. So we're learning a lot overall. And again, this is just students go out and collecting dirt. So depending on where they get their dirt from or the climate or the environment that they're getting from, we're seeing in some cases a lot of similarities, but we're also appreciating a lot of diversity in the viruses and infecting different types of bacteria as well.
Awesome. Thank you so much. I mean, obviously, if we were in person, we'd all be clapping for you. But everyone's muted, so it's not going to happen. But thank you so much for, and also thank you for, showing up last minute since our other speaker canceled, which was very unfortunate. It's always wonderful to talk to you and see you and work with you. I know you're leaving, but we'll send everyone your contact information if they have additional questions. They want to be in touch.
Yeah, if you have any questions,
awesome.
The last TEP that I did, I have several teachers contact me, and we just talked about things offline. So feel free. I'm happy to if I can.
Awesome. Thank you so much. Have a wonderful day. I know you have to go after this. All right. And with that, Frank, you're up. I'm so thrilled that you, I'm really excited to have a Boeing presentation. So wonderful.
Not a problem. My camera, okay. There you go. How's it doing, everybody? I'm gonna bring up my presentation. Before I do that, I'm gonna do a quick level set. Want to make sure everybody understands I am actually not a STEM professional. My background is in business, but I'm probably more attuned to a lot of the teachers than I would be to our engineers and IT professionals that we have at Boeing. But basically what we're gonna do, I have a little presentation with a few videos, but I really wanna pick your brains as much as you pick my brains. So what I do has direct impact to the K through 12 community in the region of Boeing that I support. And to keep you guys interested, since I know it's late, I have these prizes, Boeing swag stuff like watches and lanyards and mugs and stuff like that I plan to give away at the end of my presentation if you answer some questions that I have based on my little presentation. So pay attention. You might win a few items of Boeing swag. So I'm going to bring up my little presentation. Make sure I turn my sound on. Everybody can see me?
Do you want to go full? Do you want to put in presentation mode?
Yes.
Perfect. And
everybody can see?
We're doing great.
Okay, let's kick this off. I like to kick things off with the video. One of the things I found out about most people, they have these perceptions of Boeing that we just build airplanes, but it's a lot broader than just building airplanes. I'm working in the Boeing's South Carolina facility in North Charleston, South Carolina, which is right next to Charleston, South Carolina. So let's kick this off with this nice little video.
Ignition sequence start. 10, 9, guidance is internal. 6, 5, 4, 3, 2, 1.
One of the reasons I like to start that video, it gives you just a little snapshot of the vastness of our company. And that's just a snapshot of what we do in South Carolina. This is a little bit about me. As I said, my name is Frank Hatten. I'm actually a program manager in our global engagement organization. I have oversight for all of our K through 12 engagements for the state of South Carolina. So anything in that space involving school kids or the community that involves that community, I have oversight for all of our programming. So I'm listed as an education relations specialist, but my background is basically a program manager. I'm not originally from the South Carolina area. I was born and raised in a small community in Alabama called Tuskegee. It's a small rural community, about 10,000 people. From a logistics standpoint, it's about 30 minutes outside of Montgomery, Alabama, the state capital, about 15 minutes outside of Auburn, where Auburn University is located. It's sort of smack dab in the middle. Married. My wife is from South Carolina. We met in grad school. I have 2 sons. Both our sons went to Tuskegee also. So they're the 3rd generation of my family to attend Tuskegee University. From an education standpoint, you see I have a few degrees. All my degrees are in business or in program management. My 2 undergraduate degrees are in business administration and business management. And the degree at George Washington University, that's a master's degree in project management. I am a certified PMP, certified project manager, and have about 40 to 45 years of corporate experience. I'm also retired. My first career was with IBM. Started working with IBM after my sophomore year in college as an intern, and was fortunate enough to meet a gentleman there who became my mentor and worked with me to help get scholarships so IBM could help me pay for school. And I ended up interning with them the next 2 years and had a job waiting for me when I walked out. My career with IBM allowed me to travel around the country in different parts of the world. I ended up my career with IBM being stationed in Charleston, working out of my house and traveling as a program manager for our point of sale division. So if you ever go into a Walmart or a Sam's, you see the cash registers. I was over the team that got those cash registers, the IT portion of those cash registers in Walmarts and Sam around the world. So I knew Walmart from the time they started back in Arkansas till they started blowing up. And then transition out because I was a little bit burnt out of corporate America. And I actually wanted to be a teacher. But believe it or not, when I retired from IBM, there weren't too many openings for people who had a business background to go into the public school systems. I was told I had a STEM-based degree in math or science. I could walk out and walk into any school in the state of South Carolina to get an opportunity. But with all my experience, all my degrees, and all my willingness to get a job in education, they weren't looking for anybody with math background. So I turned to Boeing. I had heard that they were in the area, so I told myself, well, let me find something to do while I'm trying to figure out what I'll do with my life. I actually went back to school. I went to the local technical college. That's why you see Trident Technical College right there. And I retrained myself to be an aircraft mechanic. I had never worked with my hands before, by the way. I had always been in management. So my first job at Boeing was as an aircraft mechanic building airplanes. I never told anybody I worked for IBM. I didn't even tell anybody I had a college degree. I started there on the production floor, building airplanes, turning wrenches, drilling holes, putting in fasteners. That was my introduction to the world of aerospace and the world of Boeing. Because of my background in project management, one of the things that I started doing, I would rewrite their processes because most of their processes I thought weren't very robust. And because I was accustomed to working more than an 8-hour job, I would just hang around after I got off work and break into their computers and rewrite things. Did that for about 6 months, and then I said, let me go talk to somebody about how we're building our plane from a process standpoint, not an actual build standpoint, just a process, and see if I can help them out. Because a company like IBM Boeing have open door policies, I was able to get a meeting with our then vice president of our Charleston facility, and we talked about what they were doing, and I shared my background. Before I left that meeting, he offered me a job to run his business office, which I had one stipulation as far as whether I would take the job or not. I say I take the job as long as I don't have to travel. I traveled so much at IBM, I didn't want to travel anymore in that part of my life. Plus my sons were hitting that middle school, high school stage, and I figured I needed to be there for my sons versus having my wife trying to manage two young, very energetic young men. So I accepted a job as a program manager for our Boeing South Carolina business office and managed all of our inbound statement work for all our partners that brought parts into our facility from around the world. So basically I negotiated contracts with customers and suppliers from all over the world before we could accept their parts that they were sending in for us and were building for us. I did that for about 3 to 4 years. And then I got an opportunity that I thought was built for me. I got an opportunity to develop a program for outreach for our facility. The way I got that job was kind of interesting. I'd always had this passion for volunteering. So I was that guy at IBM that always asked people, let's go down to Habitat for Humanity and help build houses. Let's go down to the soup kitchen and let's feed the homeless. Let's go to the schools and read to the kids. I was that guy. And when I couldn't get people to do it, I just do it by myself. So I developed a reputation around the community as that person who would come in and just help out, regardless of what the need was. So when Boeing wanted to develop an outreach program to teach young people in the South Carolina corridor about aerospace and advanced manufacturing, they called upon me to see if I'd be interested in helping develop the program. And once again, my only stipulation, I don't want to travel. So I took that job, and they asked me to develop an outreach program for South Carolina. The mission of that program was to engage the community, to teach them about not just Boeing, but about this thing called aerospace advanced manufacturing, STEM, which we found out that so many people in that corridor just didn't have the breadth of knowledge of. And we were having some very difficult times hiring people from South Carolina that had skill sets that we could bring into our facilities to take our jobs. These are entry-level jobs that just require a high school diploma, but also require you to have some basic skill sets like math skill sets, science skill sets, reading comprehension skill sets, those skill sets that you automatically get as you study STEM-based disciplines. So I developed the program from scratch that we're going to talk about in a minute. The other component that I like to share with people about what I do is the volunteer piece, my hobbies. It's surprising that at this stage of my life, and I'm in my 60s, that I'm getting paid to do what I would do for free, what I do for fun. I'm heavy into volunteering. I'm an Eagle Scout. Both my sons are Eagle Scouts. I've been in scouting since I was, 6 years old. And through scouting, my sons are both certified scuba divers, certified sailors, and they learned to fly airplanes at summer camps in Tuskegee. And they did all that before they got out of high school. You see the photos of my family at the bottom. The picture in the middle, that's sort of what I look like most of the time, as I have to go around the state and talk to people quite a bit. And some other pictures are pictures of me talking to kids at after-school programs and gymnasiums and things like that. So that's a little bit about my background as far as where I came from and how I got to Boeing. Now, a little bit about Boeing, just in case you don't know a whole lot about the company before I get into some stuff about our programming. Boeing is over 100 years old. Most people don't realize that. Founded in 1916 by Mr. William Boeing. That's him right there with holding the bag in front of us. It is the largest aerospace company in the world if you look at our major components. The commercial side, most people are familiar with that because if you fly on airplanes, that's our commercial division. Most people are familiar with our defense division because they see we have fighter jets and things like that. But people aren't as familiar with our space division. That's been working with NASA since the conception of NASA. Every time there's been a space launch, there's been a Boeing component associated with that space launch. And most people aren't familiar with our satellite division. Our satellite division is the second largest satellite producer in the world. And our global services division is a division that works to fix the parts and the things that we sell to the world. So 4 very distinct positions and divisions that make us the largest aerospace company in the world. We presently have about 165,000 employees worldwide. And in the Charleston facility where I work in North Charleston, we have about 6,500 employees. We're the 3rd largest employer in our region. And we've only been in South Carolina for right around 11 years now. So we have a pretty strong footprint and strong presence in North Charleston right now. So with that, let's talk about my program. The name of the program is called Dream Learners. Why do we call the program Dream Learners? Well, in Boeing, South Carolina, we have an airplane that we build called the Boeing 787 Dreamliner. So it was a play off the name of the airplane. So Dream Learners, Dreamliners. As I said, when we first developed the program, it was mainly developed to get the community a better understanding of what we were doing in South Carolina from a Boeing standpoint. We talked more aerospace. We talked more about legacy Boeing and things like that. But as we developed the program over the years, we changed the footprint of the program and changed what we're trying to do and made it more of a STEM-based program with a mission to inspire more youth to consider going into STEM-based careers. So we talked just as much about Google or Amazon or BMW or Volvo, and all these companies are in South Carolina, as we do about Boeing, because STEM is STEM no matter what you do. And most students and most parents in the community that we serve don't realize how much STEM they're involved with on a day-to-day basis. They assume that if you're not an engineer or an IT professional or something like that, you're not doing STEM. They don't realize the day-to-day things they're doing in STEM. And once you break it down to that basic level, it begins to open up doors that a lot of people don't realize they can step through. And our programming starts in kindergarten. A lot of people told us that was too early, but if you look at statistics, if you're not involved in these things by the third grade, you're usually behind the 8 ball a little bit. So all of our programming starts in the 3rd grade. When I say programming, our programming involves an on-site program and a program where we actually go into the community. Since COVID hit in March of last year, we've converted our entire program to a virtual program. So everything we do is virtual, so we can basically do our program with anybody in the world if we chose to. But for right now, our target audience is the South Carolina corridor. So we primarily just support the South Carolina corridor unless our Boeing facilities around the country ask us to assist them also. We base our programming on our Boeing resources. And this is something that you guys might want to investigate. Our Boeing Future You and Curiosity Machine resources are free STEM-based resources that are grade level specific, that are approved to national standards that anybody can go and access. It has step-by-step STEM-based activities that anybody can do in the classroom or they can do at home. We use those activities and those programs to work with the community and work with students and educators to introduce STEM to them. And it's been extremely successful. Since the program started in September 2012, we've reached over 715,000 students as part of this program. That includes about almost 65,000 students coming on our property for our on-site program. That's an educational program that's approved by the state. We're the only company in the state of South Carolina that does outreach programming to every county in the state. I personally, as part of my work scope, visit every county in the state. That's 46 counties, at least twice a year to do outreach programming. So it's been very successful. I won a lot of local, state, and national awards, and it's in the process of being duplicated at other locations across Boeing. So please consider, if you have a Boeing facility in your area, reaching out to your Boeing counterparts in your area to see if they have something that they can offer in your area. I think I was told that most of you guys are from the Maryland, Baltimore area. So the closest Boeing facility to you would be the Philadelphia facility. And we know the people there also. So you can definitely reach out to them about their outreach programming, some of the things that they're doing. And this is basically what we're offering in our programming. Pre-COVID, our on-site career speakers sessions where we'd actually go to the schools, or go to the churches, or go to a community center and do on-site career speaker sessions. We do the same thing by doing STEM activity sessions, using those base offerings that we have in our Future You site and our offerings that we have in our Curiosity Machine site, where we do hands-on STEM activities. That starts kindergarten up to 12th grade. In our on-site educational program, where we bring groups on our property for an hour-and-a-half long educational program that's approved by the state that meets math and science standards for force and motion, 5th grade level through 12th grade level specifically. Those were the programs we had before COVID hit. Since COVID has hit and we don't go physically to locations anymore, we converted everything to virtual. So we have virtual career speaker sessions now. We have virtual STEM activity sessions now. where the educators or the community partners will select the activity. Our team will call into your virtual session, and our engineers, IT professionals, or our volunteers will walk your students through that entire exercise, while at the same time providing information about their careers and answering questions like that. In all of our virtual sessions, we always have a nice, friendly, competitive component in the back end where we ask the students questions to reinforce what they've learned during their sessions and give them Boeing swag and Boeing prizes. Just like we're going to do it with you guys in a few minutes. We're in the process of developing an online version of our Dream Learners program, the on-site program that has a tour component, has a sit-down STEM, a team building activity component, all built into it. And that we're hoping to roll that out the latter part of this month, the 1st of February. That'll be an online virtual Dream Learners Lite version. And we have a thing called teammate career videos that we post on our YouTube channel, where our teammates, that's what we call our employees, will create 2 to 3 minute videos and talk about their careers and their path to getting into Boeing. That's been very successful since we're in the virtual environment, with a lot of people having virtual career fairs. Since our teammates can't call into the virtual career fairs, we share these videos with them directly with the schools, and the schools can show them or let the kids access them as much as they want. Been very successful. The format that we use, based on feedback we've got from educators, all of our virtual sessions are no more than 1 hour in length. And it's broken down basically with a short presentation. And for the career speaker sessions, you have a mini career panel with between 2 and 3 speakers that will talk about their careers and answer questions. And on the back end, we ask the students questions to reinforce what we've done. For the speaker activity sessions, it's basically the same, except we chop the time up with a 40-minute session in the middle. It's done where we build a STEM activity. The base program we have is where we build paper airplanes. You'd be surprised the number of STEM-based disciplines you can talk about by building paper airplanes. But we do things like build robots, also build bridges and things like that, all activities that are available to be selected from our selection base. These are some things that I always ask people to think about and access. And hopefully Kim can share this with everybody also. The first website there is our Boeing.com website that tells you where you go to find out anything you ever want to know about Boeing. That's our main website. The second link there, jobs.boeing.com, that's where you go to apply for a job. We always get the question, especially from high school students, how do I apply for a job at Boeing? You apply for a job at Boeing by going to our careers website. And this is what I always recommend to educators and to parents and the students. If you think you might be interested in a career at Boeing, at a job like an advanced manufacturing company like Boeing, don't wait until your senior year in high school or your senior year in college to start looking for these opportunities. Start as early as middle school by going to our website and seeing what jobs are out there. Anybody can do this. And if you see a job that you might be interested in, then talk to your teachers, talk to your parents or your mentors or whomever you're working with and say, I'm interested in a job like this when I finish school. And they can help guide you toward what classes you might need to take, because on that page will tell you the kind of degrees, the kind of training you would need to have to make sure that once you finish high school or college, you're at least competitive enough to compete for that job by applying with the background and the skill sets you would need. Believe it or not, the only thing that you actually need to apply for a job at Boeing is a high school diploma or a GED. But without those skill sets that we were looking for, you won't be competitive enough to actually get the job. That's the crucial thing when it comes to jobs in advanced manufacturing. It's one thing to have the degree. It's another thing to have the skill sets where you can prove that you have the skill sets based on internships, your work history, and things like that. That will make you competitive. The other things that we have here, those are some of our links to some of our online content for people that are interested in careers at Boeing. Some of our mechanical engineers and things like that, some of our IT professionals. All this is free information that's available that any teacher can share with her students or parents and look at to find out how people got their jobs and kind of their work environment and things like that. One of the things that we also developed in Boeing in South Carolina, we decided to highlight STEM-based disciplines that kids plan to go to in college by creating a thing called STEM Signing Day. Just like athletes in high school have signing days where they sign and to go to colleges to play football, basketball, whatever, we do that in South Carolina for kids that are interested to go into STEM-based disciplines. So if you're planning on attending a college and apply for a 2-year or 4-year STEM-based degree, you can apply to be part of this program. We select 2 students from every county in the state, and the students that are selected are brought to the state capitol to meet with the governor and meet with the South Carolina Department of Commerce and the South Carolina Department of Education leadership, along with our Boeing leadership team and university presidents. We wine and dine them, give them a nice certificate on TV, give them a whole day to hopefully encourage them to stay in those fields. Since we started this program 3 years ago, every student that's been through that program has got accepted into that, has gotten an internship with one of the companies in the state of South Carolina after their freshman year. So this would have been the 3rd year we would have done that. Oh, this will be the 3rd year, I'm sorry. It will be done virtually like we did it last year. It was done virtually. The year before that, when we initially started, everything was done in person. A very good program that helped to motivate the young people of the state and in the community. Now, 1 or 2 other things I want to make sure you guys understand about our company and what we're trying to do. And it's very timely that we're doing this. When we first came to South Carolina in 2009, one of the challenges that Boeing faced, like so many other advanced manufacturing companies, was how do we get enough employees from the region to staff up to do the work that we need? And how do we get a very diverse and inclusive population to make sure that everybody has an opportunity to compete for some of these opportunities that we have coming to this area? When we came to South Carolina, we initially did not understand how far behind some of the communities were when it came to getting their students prepared for opportunities. We have corridors in South Carolina where students have only seen an airplane on TV. We have corridors in South Carolina where when we bring students to our facility, they've never been to a location that had a building that was more than 1 story, so they're afraid to ride the elevator. We have to walk them up the stairs to our floor where we have our event on the stairs because they're afraid to ride the elevator because they've never been on an elevator. So when you have that kind of demographic shifts in a state, there's a huge opportunity for companies like ours to invest in that community to help bring the students up to a point where they can be competitive enough by the time they get through high school to compete for our jobs. If not, we're doing ourselves a disservice. So our programming is designed to do things like that. We also have high school internship and apprenticeship programs we developed in South Carolina, where after your junior year in high school, you can apply for a job at Boeing. We will bring you in and we actually will give you a real job while you're in school, where you work right alongside our engineers, our IT professionals and things like that. We don't allow students to build our airplane for liability reasons, but you can have jobs in those other areas. And that's been a great opportunity for us to open those opportunities up to students in the area. So that's just like a college internship, but it starts in high school. And it's been very, very successful. Quite a few of those students have come in their junior year in high school, and by the time they finish their senior year and graduating, some of them have walked into Boeing right out of high school, and others have gone into college and come back a year later as our interns. So we're very proud of that programs like that are very unique to what we're doing in South Carolina. Now, one of the things that we also do, we have slides like these. When our teammates go and visit schools and things like that, we ask them all to create little pages like this. We call one page Meet Our Teammates or diversity slides, and they can share things about their history and show the diversity of a company like Boeing. The young lady that you see right here, she's an Air Force, a former Air Force pilot, now works for Boeing. She was the first female pilot who was a mother who flew our brand new test planes. In the bottom right-hand corner, the T-7, you notice the red tails on that plane. That's an homage to the Tuskegee Airmen. She is a very nice young lady, grew up on a farm in Wisconsin. Went to the Air Force Academy, almost flunked out, but she stuck with it and became a fighter pilot. That's one of our teammates. This young man right here, South Carolina State graduate, one of the few engineers from that corridor that's born and raised in South Carolina. Has been a huge advocate for giving back to the community. One of our, what I call super volunteers, to go back to the community and try to help. This young lady right here, she's a Native American from the Oklahoma area. She said she grew up on a reservation her entire life, but now she's has a doctorate. Well, I wanted to show you guys that just to show you some of the diversity at our company.
So we have a number of questions. How, if a teacher was interested in getting a visit to a classroom virtually, how would they do so?
That depends on what regions you're in right now. I can speak to the ones that are in South Carolina region, but if the majority of the students here in the Baltimore, Maryland region, that would fall upon our Northeastern region set. So what I would say, they can email me or Mike, and we can put them in contact with our representatives in that area, and they would respond to that question. So you have my information that you can share with everybody, right, Kim?
I will. Yep, I do.
And they can just email me directly, and I will get that to our representative for your area.
Wonderful, thank you.
Now, what some of the educators on the phone might want to do, and they might have missed this when I mentioned it, our Future You and Curiosity Machine site. Those sites are open and free to anybody. You can go to those links right now, look at the content, and you can use that content in your classroom right now. Every exercise on that site has step-by-step instructions on how to do the activities. It even has a video and parts lists and everything. So you can either do it yourself, or once you email us and we identify the Boeing representatives for your area, you could potentially reach out to them to see if they would have bandwidth to have a Boeing representative help you with that in your classroom. But that's something you can do right now.
Wonderful, thank you. We had a teacher who they asked, I may have missed this because I joined a little late. Are there programs for high school students up in like the Maryland, Baltimore region, like the one in South Carolina?
I can't answer that one, but I can make sure to get the person who can answer it. By just tell that educator email me, and I will put her in contact with him in contact with the right person.
Okay. Yeah, and I should probably just email you about that, so I can just share that information with the teachers.
Yes, that and from this standpoint, that information and the information about Future You and the Curiosity Machine are two of the most important things on this call, and the links to our Boeing.com and Boeing job sites will probably be the most crucial information from this presentation for the students, for the teachers here.
Absolutely.
Now, if you're ever in the South Carolina area, give us a call. We'd love to invite you to come to our facility. We love teachers, we love educators, because without educators, none of us will be here. My mom was a retired teacher, so I love teachers. Now, let me close by saying this, guys. I love my teachers. I just adore my teachers. I think everybody always forgets that if it wasn't for teachers, none of us would probably be where we are. And I think as a country, we found out how important teachers are with what we're doing with COVID. So please don't give up on our students, especially the ones that don't get certain opportunities. We have so many students that if they just get the opportunity and get the right person to push them a little bit, it can change their lives.
Support from companies like Boeing, and especially Boeing, allow us to bring you these events and resources for free. So anyways, wanted to make sure I spent a moment in thanking Boeing for their support.